Look at the precision of modern targeted therapies. We are mapping genomes and engineering custom proteins. But if you walk onto the factory floor where most of these medicines are actually made, the process looks surprisingly similar to how we baked bread 100 years ago. This is batch manufacturing. As you can see in the top half of this diagram, the process is broken into isolated, time-delayed steps. You fill a massive vat, mix it, and then stop entirely at the 2-hour mark to wait. That weighing period is the critical bottleneck. The material sits in a holding tank while samples are sent off to a laboratory for end product testing. The entire production line sits idle for days, sometimes weeks, until those lab results come back. And the stakes during these holding patterns are incredibly high. If the lab detects an error or a slight chemical deviation from a sample taken at the end of the line, the entire multi-ton batch goes straight into the incinerator. Scaling up this system means building bigger vats and larger holding warehouses. It requires a massive physical footprint that locks companies into rigid, centralized facilities that take years to build and are difficult to modify. This physical inflexibility is exactly why national drug supply chains are so fragile. A disruption at one giant overseas facility, whether from a storm, a shipping delay, or a quality failure, instantly cascades into shortages at local pharmacies. The solution is to remove holding tanks entirely. The bottom half of this diagram illustrates continuous manufacturing. Raw materials are constantly fed in, and finished product constantly exits, nonstop, at every time interval. Looking at the data, the efficiency gains are staggering. This comparison shows that continuous systems require 70% less physical space than batch factories, and because testing happens on the fly, total quality release time drops from 30 days down to just 10. We can see how this space is saved in this schematic. Notice on the right how loss-in-weight feeders directly pipe raw ingredients straight into the mixer, which flows immediately into the tableting machine. There are no breaks and no intermediate storage rooms. By integrating separate steps, the factory floor shrinks. A process that used to span multiple football fields is compressed into a single, high-speed, enclosed engine. But a continuous line creates a massive engineering challenge. If the material never stops moving, how do you verify it's safe without pulling it offline for a lab test? Engineers solve this using process analytical technology, or PAT. Instead of a separate laboratory, specialized sensors are built directly into the pipes and mixers. They use near-infrared spectroscopy and light to monitor the chemical composition of the drug constantly, in real time. To interpret sensor data, the system uses a residence time distribution model. This schematic maps the physical journey. The RTD calculates travel time from blending to tableting. This perfect traceability means engineers know the exact location of any spec at any millisecond. If a PAT sensor detects an error, like a temperature spike, the RTD model knows exactly when that flawed powder will reach a certain point. A mechanical valve snaps open at that millisecond, diverting only the bad material down a waste chute, while the rest of the line continues. Combining these real-time sensors with strict mathematical modeling creates a machine that corrects its own errors. It isolates defects down to the gram, completely eliminating the risk of a multi-ton batch failure. You could build the most advanced continuous factory in the world, but if every government health agency requires different documentation and safety proofs, you can't actually sell the drugs internationally. That is where ICH Q13 comes in. It is a globally harmonized regulatory guideline agreed upon by agencies like the FDA in the US, the EMA in Europe, and the PMDA in Japan. It acts as the unified rulebook for how to safely develop and validate a continuous manufacturing line. By operating under Q13, a manufacturer can design a control strategy once, knowing it will meet the safety requirements across different jurisdictions. This removes years of redundant paperwork and accelerates the timeline for new drug approvals globally. ICH Q13 is the catalyst that turns a theoretical engineering marvel into an actionable, worldwide industrial standard. It gives companies the regulatory certainty they need to finally invest in and build these systems. With a unified global standard and equipment that takes up 70% less space, pharmaceutical companies are now deploying modular factories. Instead of building massive structures from scratch, they can drop highly efficient, pre-built, continuous manufacturing units directly where they are needed. These high-tech, smaller footprint plants can be built domestically.
Companies like Janssen have already successfully transitioned major products to continuous lines in facilities like this one, pulling production back from vulnerable overseas networks. The roadmap is clear for the rest of the industry. Analyze your process dynamics, install real-time PAT sensors, and follow the Q13 guidelines to validate the system. Replacing the giant chemical vats of the past improves factory efficiency and ensures that life-saving medicines remain available exactly when patients need them most.